Hello, everybody. Welcome back to Creation Myths. We are live, at least that's what StreamYard is telling me. So I'm going to wait just a moment, as I like to do, make sure I can hear everything on YouTube. Once I know that we're good, we will officially get started. All right, it sounds like we're good over there. So if there are any audio, visual, or otherwise tech issues, let me know in the chat. I'll keep an eye on it. I will see if anyone indicates that there is a problem. Otherwise, we're going to get into it right here. So this is one of our conversations about evolution and creation, and my guest tonight is Glen. As you can see on screen, Glen is a frequent commenter on the channel, and I've repeatedly made the offer to commenters that disagree with me. Say, look, you have substantive disagreements. Shoot me an email. Here's the address. Let's talk about it. Like, we can set up a conversation to talk about it. And nobody had ever taken me up on that offer until Glen did. So we, back and forth via email, and we set this up. And so now we are going to talk about it. So Glen, like I said, commenter on the channel, interested in science, has had questions about evolution and evolutionary theory for a while. He does have a degree in microbiology with a minor in biochem. Correct me if I'm wrong about that was.
Correct.
Did I get that right? Got it right. Okay. So that's where we're coming from. So Glen has questions, and I'm going to try to answer them, and we're going to have a conversation about it. So let me just send it over to Glen. Before I do that, let me just take one brief moment to say, so Glen is the commenter on the channel that you may have seen in the comments. He goes by the username Crackhead Biden. And I will say this, he has managed to stay just on this side of getting banned, because as everyone knows, I have an extremely sensitive trigger finger when it comes to banning people for being rude and name calling and such. So I will say this on air for everybody to hear. Glen, you are on the tightest of leashes for this conversation, and if it goes even a little bit off the rails, I will not hesitate to turn this car right around.
Wow.
And with that, let me turn it over to you. Feel free to introduce yourself further, introduce what you want to talk about, and let's get into it.
Well, first of all, I promise this is going to be cordial. And most of the stuff I do online, you got to kind of push some buttons to get a reaction, and here we are. So I'm not an ass of a person, to be honest with you. So I appreciate the opportunity. So like I said, when I went to school, I had all these questions about evolution when we got into the cellular biology and things like that no one ever could seem to answer. And so I've always carried them along, and I've never had them answered. And so I thought this would be a great opportunity to talk about them. I'm open-minded. I don't come from this from any kind of a religious point of view whatsoever. I think there's only two options. Either there's spontaneous life that evolved into all complex life, or there's creation. So if you can't prove evolution, then I guess it falls to the other one. But we'll move on from that. I think when I talk, I wanted to just make sure that when we're using the words that we're clear on what I mean by them, okay? And so in school, when I was, the word information for me, what I was always taught, it was just simply, it is just a portion of DNA that codes for a functional protein. That's it. It's just that's all it means. And then and I know there's a micro and macro argument about evolution and all that, but I'm just going to stick with evolution from a common ancestor, which means a first simple cell had to add a bunch of DNA to become all complex life. And then I'm just going to refer to the stuff we see every day now as adaptation, speciation, and whatever that means. I think that keeps it simple. I guess I'll just start with my first thing. I don't want to get into abiogenesis. I don't think that's worthy of anything anyway. It's just sort of a guess.
Good. Well, so it's funny you should bring that up, because I'm actually teaching that in class tomorrow. So it'll actually be a really good time to bring it up. But I'm not a biochemist, so I am not the best person to talk about that. So I don't like to talk about abiogenesis either, just because it's way outside my area of expertise. I don't understand the biochem as much as I could.
And so I think when we start, I think it's important to start from the very, very beginning, and we start with early life, right? And so the research has shown that the simplest living cell capable of cell division requires 473 genes. That means proteins, metabolism to power it, functional cell membrane to move things in and out to regulate the inside of the cell. And that's all required before evolution can even begin. And so that to me seems like a big, big problem, because how do you build that complexity if you can't use the evolution to do it? And then if you look a little further into that, all living cells have DNA, proteins, and ribosomes. But here's the problem is proteins are required to regulate the reactions to make functional DNA. DNA is required to code for those proteins. And ribosomes, which consist of roughly 60 or more proteins themselves, are required to make all of those proteins to begin with. So you've got three things that all are required together to make each other. So how you come up with that gradually over time with evolution, that's another puzzle for me. And I don't know if you want to stop there and talk about that.
Yeah, we can absolutely talk about this a little bit. So this sounds to me like an irreducible complexity problem applied to the first cells, basically. So in order to be a functional cell, something that we would look at and go, that thing is a cell. It has all the components of a cell. It functions as a cell. In order to do that, you need a number of components that are all interdependent on each other. So how could that system, that cell, have arisen through biochemical processes, evolutionary processes, whatever, when the functional components it needs can't function without the other components already in place? Is that a fair representation of kind of the problem that we're stating?
Correct.
Okay, so the first thing that's going on there is you said that you can't have evolution before you have those first cells, but evolutionary processes definitely predate cellular life. That's like 100% the case, and we can demonstrate that experimentally, that you don't need actual cells in order to demonstrate things like selection and replication occurring. So as long as you can have a population that has variation, and then some sort of growth in that population where there's varying levels of success, that population is going to evolve, and you don't need cells to have that system happen. So that's the first thing, is that you don't need cells for these processes to operate, right?
So that kind of gets to my point with the evolution part, is there is no answer how it happened. I mean, there's a guess, and there's an assumption that things evolved and then they created themselves, but there's no clue how it could happen. And then the other thing, one of the things that you hear is, well, there was an RNA world and ribozymes are self-replicating molecules and they but there is no example of a self-replicating ribosome, none. And a ribozyme, because of the chirality problem, it cannot form spontaneously in nature. There's like 60 plus different ribonucleotides in a ribozyme. Each one has a possible chirality of 16. So you got 16 and you got 60 different times. That puts an astronomical number to make it impossible for it to form. Plus, the chirality constantly changes. And so that's fixed in a living cell by enzymes. Mm-hmm. So even if you were to get a perfect string all the way across, it's still going to keep changing and become non-functional, because life has to have that one homochirality all the way across it. So ribozymes are not an option. Plus, a functional ribosome is folded and not straight. So you can't line them up and duplicate a folded ribosome. So that whole that whole thing is sort of non-starter.
So we are talking about abiogenesis, and that's totally fine. Happy to do it, but that is the topic we're talking about.
Yeah, I understand, but what I'm saying is we got to the point where you have to have a living cell to begin what we know as evolution. That means a cell copying a perfect copy of itself, duplicating with slight mutations, creating a population, and creating complexity. So we got to start with the first living cell, and somehow you have to get there.
Right. So are you familiar with the, yes, and I thank you for the comment. I know there's a volume difference here. Let me see if I can, tell me if that has addressed the volume difference, if that's any better, everybody. And Glenn, can you still hear me okay? Is that all right?
I'm hearing fine. Yes, thank you.
Okay, cool. So people watching, tell me if there's less of a volume imbalance here. I don't have any fancy software or anything, so I don't have as many tools as some other people do here. Okay, so this is just, now we're going down the list of abiogenesis topics. So chirality is one that you pointed out. Are you familiar with the literature in terms of the mechanisms for generating homochirality within a pool of chemicals? Because there's a lot of literature on it, and there's a number of proposed mechanisms that have been demonstrated.
Yeah, I've heard it, but it's not something that is done in nature, if you will.
So you've heard of it, but have you looked into it? Have you read any of the literature?
Here's the thing. I didn't want to get into abiogenesis. The only reason I brought up the ribosome was because a lot of people say that's an easy way to get there, but it's not. That's the only reason I brought it up.
I agree. It's not an easy way to get there. For just so everyone in the audience is clear, there's a number of proposed solutions for the homochirality problem. One is called autocatalysis, where you basically have a neutralizing reaction between the left and right-handed monomers paired with a slight imbalance. That leads to kind of a cascade of selection where you end up with an increasingly homochiral solution, and that reaction is ongoing. So it fixes the problem of kind of constant the movement towards equilibrium in the solution. You also have polarizing radiation is capable of converting one to the other. So you end up with polarizing radiation. You end up getting a monochiral, or at least an imbalanced pool. And remember, you don't need monochirality or homochirality in order to have functional molecules. You just need to have enough of an imbalance that you get function using one orientation, and now you're selecting for that orientation. And this goes back to the idea that you don't need cells to have evolutionary processes. Once you have more stable molecules that require being either right-handed or left-handed, then you're selecting for those things because that orientation becomes inherently more stable. So you end up shifting the equilibrium towards one side. And it's a positive feedback loop where you end up enriching in one orientation over the other. This has been demonstrated experimentally. It's not like this hypothetical thing that we think may have happened. These processes can be demonstrated experimentally.
Yeah,
so that
if I'm not mistaken, those were amino acids. So they had a left and a right. And so
that's in, I believe those were done with amino acids. Yeah.
Yeah. So you had a left or a right, two options. This says 16 times 64.
Right. With nucleotides, it's harder because there's more pivot points where you have to have it lined up. Yeah.
There's been no experimental data that's ever shown that's possible. Not with a ribozyme.
Well, with a ribozyme, no, but with nucleotides, yes.
Yeah,
you can get an enantiomeric imbalance with nucleotides. That's been demonstrated.
Yeah. But it's the ribozyme that's the tough one.
Well, there's other experiments that have shown that you can generate ribozymes just through random polymerization. That's also been demonstrated experimentally.
The only one that I know of, they took a homochiral ribozyme, they split it in half, they put it in with a mixture of a fully formed homochiral one, and then they just sort of linked them together. They lined up, and then they called that an auto.
I think you're referring to that's the Joyce kind of T-shaped thing, right? Yeah. So that's not the only one that's been done at this point.
What do you say we move on from that?
Okay. Yeah, that's the Joyce famous ribozyme. It's not the only one that's been done at this point, but also just a point on just experimental design. You're necessarily going to put it in a homochiral mixture because that's a controlled experiment, right? You're not going to introduce other variables into that experimental design. If the question is, can we polymerize something to make a sequence that does a thing, right? A ribozyme or whatever. Can we randomly polymerize and get a ribozyme? Yeah, you're going to design that experiment so it's a controlled experiment. Right. It's going to be, it's going to be a homochiral mixture. That's not a point against the process. That's if you can show one way experimentally, okay, we can generate an imbalanced mixture leading to homochirality. And then we could show another way, given homochirality, we can have random polymerization that leads to a function, right? You can imagine it would take a really long time to string those two things together. So we break it up experimentally into controlled experiments showing each piece. That's the point of why we do it that way.
Good enough.
Okay, so what's the next thing?
So anyway, I think we've run across this where there's no answer, right? There's no answer one way or the other.
I mean, I think there's an answer. I'm going to give a 2-hour lecture on this tomorrow on abiogenesis. We're going to go through a lot of stuff where we have answers. There's the problem with abiogenesis and like getting ribozymes and getting cells and stuff. Frankly, the problem isn't that we don't have plausible pathways. The problem is that we have experiments that demonstrate multiple plausible pathways, and we don't know which one is the most likely.
There's not one single hypothesis of how a ribosome, DNA, and proteins formed. Not one.
There's not one hypothesis for how these things formed?
Not that I've ever heard. That is the one thing. The ribosome. The ribosome. The ribosome. Yeah, the ribosome. The ribosome, DNA, and proteins, how they formed all together in one cell. Spontaneously. There's not even any, I guess. No one has one.
I mean, we've experimentally generated protocells with RNA polymers inside them.
Yeah, but a protocell is just basically a lipid bilayer with nothing going in and out. It's just basically a tune for things to degrade in.
Well, it does have things going in and out. It's selectively permeable. It's not impermeable. And what you just said is that we have no hypothesis at all, but like we've done this stuff in the lab and we do have hypotheses, multiple hypotheses. You've got the protocell hypothesis, which could form either kind of surface of waters or it could form catalyzed by reactions at deep sea vents, which is kind of a newer idea, but it's taking off. Then on the complete other side of the ledger, you have all the metabolism first hypotheses that have the reactions happening first and then becoming kind of contained later, compartmentalized later, but you have the reactions happening among biochemically active geological formations like deep sea vents or tar pits or something like that. Tar pit's a bad example, but like hot springs, where you have lots of chemical activity, lots of gradients of different types of chemicals, and you can have reactions involving acetate going back and forth between oxidized and reduced states, and these reactions become compartmentalized later on, right? So it's a metabolism first rather than a structure first approach to building cells. Again, to say we don't have any workable hypotheses for this doesn't strike me as realistic. I'm literally teaching a class on this tomorrow, and we're going through the hypotheses and the evidence for and against several of them.
Okay, let me be more specific on the hypothesis part. DNA can't form in nature by itself. Proteins cannot form outside of a living cell on their own. And ribosomes can't form on their own either. So there is no hypothesis on how they actually formed inside of a cell altogether. And all the vents and all that other stuff, abiogenesis and how a protocell, that's all separate. I'm talking about these three things.
So how do these components come together into a cell?
Form, create themselves.
Well, so I think they form, I mean, goodness, we could go through we could go through the process. I mean, I think they form through chemical reactions in the environment where you have smaller carbon-based molecules that polymerize due to energy absorbed from the environment. So they reach more stable states as they absorb energy from the environment. And like to say that we don't, do we know how
No,
this happened? No.
no, exactly.
No.
That's
Do
all.
we have,
That's all we need
I,
to do.
we don't know how gravity works.
I know. Well,
Like
that's
it's still, we know that gravity exists and we can measure it and quantify it and like make predictions based on it, even if we don't understand like how gravity works, right?
what
In
we.
the same way, do we know how DNA formed initially? No. Do we have a number of like lines of evidence that show potential pathways for it? 100%. Absolutely. Also, it's worth noting that DNA doesn't have to form at the same time. It's the consensus is an RNA peptide world at first, whether that's within cells or outside of cells is open to debate, I think. I kind of have come around to favoring the metabolism first side of things. They've frankly convinced me. But you don't need DNA at first. All you need is RNA and protein. Uh, and with them reacting together, then DNA comes later. And the whole like evolution of why DNA instead of RNA and why thymine instead of uracil and all that stuff comes later. You don't actually need DNA first. You need RNA and peptides.
So I have everything I've read. Um, most research now is throwing out an RNA world altogether because RNA is just too unstable.
That's what the peptides help with that. Yeah.
You know, but then you're saying you need a system built then before you can keep the RNA together. So now you're in irreducible complexity.
No, you need other stuff in the environment is all you need. And
but, you know, RNA reacts with itself. It has a shelf life of about 2 days. That's about it.
It depends on what it's interacting with. And we've shown experimentally that you can form the RNA bases and you can polymerize RNA and it's sufficiently stable to be enclosed within protocells. Like,
and how do you store RNA in a lab? What do you have to
really
do?
cold, make it really cold.
Exactly.
Mm-hmm.
Because if you have it at anything above the temperature that you would store a Pfizer vaccine, it degrades in no time at all. So it cannot be a substitute for DNA.
That is only if it is biochemically inert, if you're keeping it in a solution that's not doing anything. But if you're working with like RNA viruses, you don't have to do that. The RNA viruses doing their thing can go in an incubator and it's not going to break down in 2 days. Those viruses are going to grow and be super happy. So it's only a stability issue if you assume that it has to form on its own with nothing else interacting with it.
Well, that would be the early environment with water, right?
Why would there be nothing else there? There's going to be lots of other things, and there's going to be metal ions that can stabilize things. There's going to be other molecules that are forming. Anyway, we're spending time on abiogenesis. We don't have to do that, but I do want
yeah, go ahead. Sorry.
I do want to say this comes across as the irreducible complexity argument applied to the first cells, which strikes me as not valid, aside from the abiogenesis side of it, because irreducible complexity itself is not a valid argument for the simple reason that no matter how you define irreducible complexity, we've directly documented such systems evolving, systems that meet those criteria.
Okay. So what I gather from this is there's a lot of ideas, there's a lot of guesses, there's a lot of proposals, but there's no actual evidence that you can see, observe, test, or anything that shows where DNA RNA they came from. Where did the ribosome come from? There's nothing. I've never found it. That's all I just wanted to say. That's one thing.
Okay.
Right.
Well, my class tomorrow is going to be really short then.
I have watched over and over the PhD guys from Harvard doing abiogenesis from first replication all the way to production of proteins multiple times. So I'm familiar with what I'm saying. So I know that it's all a lot of guesswork. Okay. So my second one is, and we'll get the evolution part, get out of that, right? I appreciate you being patient with me.
Oh, I'm good.
So the other part I had with it is evolution building systems. Now you're going to call this irreducible complexity, which I'm sure it is, but it's, and I've seen your talk with Dr. Behe, and I know you used the virus as an example, but I wanted to bring it up a level of complexity, right? And so I wanted to use as an example a motor protein system in a eukaryotic cell. And so people aren't confused, I'm not talking about the transport proteins in the membrane, but a motor protein system, right? I'll quickly just describe it. So in the eukaryotic cell, they're so large, they have to have these transport systems in order to carry cargo all around, remove waste and all that. So that motor protein system is basically looks like a person, if you will. It has arms that carry stuff and legs that walk across a motor tubule to deliver things. So here we have a system where you've got the arms that are a different set of proteins, the legs that are a different set of proteins, microtubules a different thing, and then the whole system only works if it's powered by an ATP system. So none of these things do anything without all the others functional. So how do you build 4 different things gradually when none of them do anything until everything's completed? Using evolution, right? So that's one of those things I just don't understand.
So I don't know specifically, I'm not familiar specifically with that system. I know of the proteins. You're talking about myosins and kinesins, right?
Correct.
Yeah. So I'm familiar with the proteins. I know their basic structure, what they do. Go ahead.
How about we just make it real simple? Let's take an eye, right? Everybody talks about an eye, a functional modern eye, right? It's got the eye, the optic nerve, the brain.
Cornea, lens, something to regulate lighting.
We don't even have to get that, right? So you always see the eye is so easy to form by evolution. They'll show the light sensitive cell folding and creating this and that, and they have it actually in their diagram sitting in an optic nerve. So the part that evolution I don't think can do is that eye, you can fold it all you want, but it does nothing. That does nothing until it's connected to an optic nerve to transmit something to the brain. So without that transmission, there's no reproductive advantage to keep building it. The optic nerve doesn't do anything unless it's connected to a functional eye.
Okay, so
not only on top of that, you have to have developmental genes, a third system that know to only put that new proteins or whatever in those 2 spots where the eyes go, and know where to build that optic nerve to connect it, over to the eyes. So you got 3 different systems that all have to build at the same time, almost as if they're communicating together, which do nothing until they're complete, right?
So it's an irreducible complexity argument applied to eyes and how nervous systems and how they develop and work together. So there's a whole lot there, and I do have answers for you. So first thing is that to do this, you don't need new proteins. And this is true for a lot of systems where you have a new structure or a new function. You don't actually need a new protein. So eyes, specifically the photoreceptors, the opsins, or the proteins that do that, those are a subset of proteins called they're serpentine proteins. The smaller set is G protein coupled receptors, which are found throughout animals. And I don't even know, they may even be in other things, but I know they're ubiquitous throughout animals. And they're chemoreceptors. So they're transmembrane proteins that embedded in the cell membrane. They have a piece that sticks out and they have a tail that sticks into the cell. So they attach to, some chemical substrate on the outside, and they wiggle the tail and they get a signal on the inside. I'm oversimplifying that for everybody. They don't literally wiggle the tail, but that's what happens. And opsins are a subset of those proteins that mutated, and instead of responding to a chemical signal, they respond to photons. The rest of the protein is basically the same. It's just a mutation on the extracellular domain that would otherwise respond to a chemical signal. Now it responds to a light signal. So you don't need a new protein to do that. But you do need a way to detect that stimulus, right? You need some way of like dealing with that. You need, if you're a multicellular animal and want to like respond to a stimulus, yeah, you need nerves, but you don't need a brain. Jellyfish have what's called a nerve net. They have very rudimentary eyes, you could call them. They basically just detect light and the direction of light. They don't have a brain. They can't process any of this information, but they have this nerve net that's this decentralized kind of nervous system where it's all very almost like algorithmic. You put it in, you get a response out. If you touch it here, it contracts this part and it moves in that direction. If it detects light over here, it does this. If it detects light over there, it does that. It's just in very basic processing and out. There's no conscious processing. There's no brain. You don't need a brain to do that. You just need a way of taking that signal from one part of the body where it's detected and distributing it to the rest of the body. And there's going to be variation in how the body reacts to that. And the ones that react correctly, that's going to be selected for the ones that have the nervous system hooked up correctly. And if you have it wired wrong, you move towards the light instead of away from the light or something, then you got a problem. You're not going to survive, or you're not going to survive as well. So you don't need all of these components in order to have a functional eye nervous system situation. And once you have the basics in place, it's not hard to increase the complexity of that because it becomes a positive feedback loop. If your eye becomes more sophisticated, it becomes worthwhile energetically to have a nervous system that's more sophisticated to deal with that extra information. If you have a bigger, more complex nervous system, you're capable of dealing with the information from a bigger eye. So you get this feedback loop of complexity where you're selecting for greater and greater complexity. And we see this across lots of different animal groups because, as I'm sure you know, eyes don't all share a common ancestor. Eyes evolved independently probably dozens of times across different groups. Some of them are very different, some of them kind of the same, even though they're in very different groups of organisms. So you get the proteins, you have the basic nervous system, and then the other thing you brought up was development. And the cool thing with development is you don't need, again, a whole lot of new stuff. You're just moving levers at different times and in different places during development. And eyes are a great example that I actually use in class to illustrate this. We're going to do this next week in class, because to get like a spherical eye, right, instead of just like a flat eye spot, a pigmented spot, you don't need a whole new set of genes that say develop an eye. You just use existing genes that say fold inwards. And that's a process that happens all the time in animals. We get folds in our intestines. We get folds on our dorsal side when we do neurulation early in development. Signals that say fold inwards when we express a certain set of genes are ubiquitous throughout animals. It's a way of increasing surface area, and we love increasing surface area. So you have a surface that can detect light. You tell it to fold inwards, now you get a cup. You pinch it off, now you have a sphere, right? And these are processes that we see all over the place throughout animals, including in other places in the human body during development. So you don't need a whole new set of developmental genes to make an eye. You're just changing where you're expressing genes that are already there during development, and that's how you get it. Go right ahead.
Yeah, in that part. So that very, very first step, you said that folding, right? Mm-hmm. I don't see where that would create some kind of a reproductive advantage. Now, I would think that a mutation like that would be pretty rare.
Oh yeah, probably.
Uncommon. That would happen in one individual at once, right? And so
maybe
a little fold in something is not going to create a reproductive advantage to make that the dominant gene pool.
What if I can tell where light is coming from and everybody else can only tell what's dark or light, but my eye spot is just a little concaved?
Well, if
So
you
I
don't
can
have,
tell the direction,
but if you don't
right?
have an eye spot yet,
Well,
you
the eye
can't.
spot,
All you
eye
have is,
spots are easy.
yeah, but an eye spot doesn't happen like that. It has to take time.
Right.
Evolution happens slowly, gradually. It's not an overnight thing. All of a sudden, you got an eye spot that works, or a fold that does something
I
different.
mean,
It's going to be a gradual.
you know,
That first, everything that you develop first, that first little step isn't going to be much of an advantage.
there's like
Right?
algae with eye spots. There's unicellular organisms with eye spots. It's a
Understand.
very common thing
I
that
understand
is
that.
an advantage.
I understand it's an advantage, but I'm saying that things that didn't have an eye spot, we're talking about the
Mhm.
evolution of an eye, right?
Right.
Didn't have one before. It had nothing.
Mhm.
It may have a cell that could sense a little bit of light, maybe, or even the development of that. But just folding a little bit
Mhm.
and only picking the two right on top of the head to fold. But that little first fold, I don't believe is enough for one individual to have such a reproductive advantage to pass that on and make it in the population. That's the problem I have with evolution and these kinds of theories, if you will, is that first step of evolution has to be something enough to make it, a reproductive advantage.
It has to be a big enough advantage that it
Yeah,
propagates through the population.
exactly.
Yeah. So
Exactly.
there's two things. That I want to say here. One, the smaller issue that I want to address. I think the smaller question they want to address is like we can model that and show like mathematically what level of advantage you would need to achieve in order for a trait to do what's called a selective sweep through a population, right? To actually kind of become the dominant morphology within that population. We could do that math based on population size and whatever the inheritance pattern is for the trait. And you could go through and do the math and figure out how big an advantage do you need in order for that to actually happen. And the cool thing is we can actually then make predictions and go and check in the real world with things like, for example, there's a famous example of this along the Mediterranean coast with mosquitoes and resistance to pesticides. And you can actually like predict like, okay, what level of benefit do they need to get in order for that resistance to spread through the population, or will it not be enough of an advantage? You could also do this with bacteria and antibiotics, which is do that in the lab in a few weeks. But the bigger problem, so I bring those examples up because like, yeah, we do like, that is a question of like, how beneficial does a trait need to be in order for it to have this selective advantage that it becomes the dominant thing? Well, we can answer that question. We know the answer to that question. Like, we can model that and we can test our models against the real world. The other thing, though, the bigger thing that I've noticed a few times, you're saying things like, I haven't seen or I don't believe. And like, okay, you don't have to buy any of what I'm saying, but like, I have a textbook right here. Like, I can go chapter by chapter through this book where they're talking about experiments and field work and everything, where they go through, here's the principle, here's the theory, here's the model, here's the math, here's the experiments. And therefore, here's what we know, right? Going through this step by step. And this is not something that's unique to this conversation when it comes to irreducible complexity. A lot of it comes down to seeming like I'm playing a game of whack-a-mole, where it's like, well, how did this thing happen? How did that thing happen? I don't know how that could have happened. But the relevant question with irreducible complexity is not how any one system could have evolved, because there's always going to be a system that we haven't documented in sufficient detail to be able to say, here's the evolutionary pathway. There's always going to be something like that somebody could point to and say, that thing, how does it happen? Too many parts couldn't happen. The relevant question with irreducible complexity is it a workable hypothesis that when you test its predictions, which is like, if a system meets these criteria, that system cannot evolve, right? Is that an accurate prediction? And to answer that question, we don't have to play whack-a-mole. We just have to look at those systems and see if we can document them evolving. And like, put aside the virus stuff or the bacteria stuff, we can see those systems evolving in eukaryotes, in animals. Like, we can document the evolution of systems that check all these boxes. And I'll give you an example, and then I'll shut up because I've been talking for a few minutes. I'll give you an example of this, though. Evolving the ability to have placental gestation is an extremely complex trait, right? Let's think about all the things that go into placental in utero gestation, right? You need to have a uterine lining that is sufficiently, has sufficient capillaries to provide nutrients to the developing embryo. You have to have an embryo that has the right receptors that are compatible with the uterine lining. You have to suppress the inflammatory response so the uterus doesn't boot out the foreign invading cells from the embryo. You need to have it timed correctly so that when the early embryo is present, the uterus is receptive to it. You need to have it develop the intertwined blood vessels, whether it's a placenta or just the uterine lining. You need to have the blood vessel countercurrent exchange system between the two. You need to have all these things all lined up in the same place at the same time in order for that trait to appear. But we know there are lizards that have done this transition because we know lizards just, lizards lay eggs, right? But we have documented lizards that have true placentas. And we've actually documented lizards where they've gone from, the population has gone from laying eggs to given live birth. We've documented that transition over the last few decades. That's an irreducibly complex trait by any standard, but when we could go through, just like we did with ribozymes and ribosomes and DNA and protein or eyes and nervous systems, you can do the same thing with giving live birth in lizard populations. But
if I'm not
we got to see that happen.
If I'm not mistaken, is it 5-10% of lizards have live birth as it is? Skinks?
They do. So most lizards that give live birth do, they're oviviviparous. So the eggs basically develop internally and they give live birth. There's a very small subset that actually have a placenta. They have an invasive placenta. So they don't have the internal eggs. It's true placental gestation.
So it's possible that those genes could have been passed down when they speciated.
So they inherited those genes for placental gestation from however long ago, and all the other lizards just happen to lose them, and it looks like they have the ancestral state for laying eggs, but they actually have lost the ability to give live. That's a much more complicated hypothesis than placentas evolved.
I think that the loss of things is much easier to explain than the gain, right?
So when we lose things, we can see that though, right? Like in the human genome, we can find pseudogenes for stuff that's gotten broken and lost. Like we know what that looks like. Like for example, we can't make vitamin C, right? That's a pseudogene that we have, right? But like lots of other, other primates, they can make vitamin C. They have the full gene. We have the pseudogene that's broken. We lost it, right?
Yeah.
So we know what that looks like. So if the hypothesis is that the lizards could ancestrally develop a placenta and give live birth, only a very small subset can still do that. The rest have all lost it. In a way, they've got these loss of functions in a way that left no signs of it. No pseudogenes related to placental development, nothing. It's just like it was snapped out of their genome. Is that a more parsimonious hypothesis than maybe irreducibly complex structures can evolve?
I just, lizards and live birth is just not my thing. I'm sorry.
Okay.
So I know we talked about the eye, but how about let's do the fish leg thing, right? So there you have to add new proteins. You can't. Yeah, you do, because structure, right? A fish has layered muscle. A leg is a completely different kind of muscle. You have to have tendons, ligaments, bones, knees, joints, toes, claws, skin. That's all different than a fish.
Fish. You keep going. Yeah.
Yeah, and I was going to say, but you have to be able to build this stuff gradually, but you also have to have your Hox genes coordinating it as the new things are happening, so they almost communicating. So you need new information to the Hox genes to say, hey, I got this new thing going to be built, and you're to only put it in the fin. You're not to put it in the body, you have to put it in the fin, and then only in this part of the fin. And only after this part of the bone is built, then you can put the knee in. You know, so it's a system where the two have to communicate. And how does evolution build two separate things through mutations that communicate with each other, right? And you're going to get to a point, just like in anything there's an adaptation like this, you're going to get to a point where you no longer have a functional fin and you don't have a functional leg yet. And so those fish that don't have a fin that can swim with or support weight are going to be outcompeted by those with a functional fin. So it is actually a detriment, not a reproductive advantage. So you're going to find that in every situation where you're developing one thing into another thing gradually, right? There's always going to be a stage where it's non-functional, and evolution will not build it any further because it's not an advantage.
Right, like penguins, for example, who can't fly, but they still have wings, right?
Yeah, and a polar bear evolved from a brown bear by breaking a pigment gene and a fat metabolism gene. And
well, I was being a little facetious there.
I'm just saying that genes are broken all the time to adapt animals. That's
right. So
loss of information.
So first thing with that last point, the intermediates are perfectly functional. I mean, so penguins have lost the ability to fly, but they use their wings to swim. But they're still wings. They're still morphologically wings, but they use them very effectively to move underwater. Look at seals. They weren't what? Sorry, you cut out for a second.
I'm sorry. They weren't legs first. They've always been wings. They just became non-functional wings. They didn't.
They became
to another. They didn't need them to support their weight or walk on them, and then use them for swimming. There would be a state. That's what I'm saying. There's a transition stage between something different into something completely different, right? Arm to.
Let me grant your premise. Let's grant your premise that penguin wings were never involved in weight bearing, which is not the case. But let's grant the premise and say, okay, they started as wings.
I'm sorry. They've always been wings. They've always flapped, right? They could support weight the whole time. They've just lost feathers and became swimming, right? They've always been functional.
So what you're describing is the process of exaptation, where a structure or a trait does one function and then it changes function to do a different thing. Yeah. So like for penguins, there was a point where they lost the ability to fly in their evolutionary history. Their wings could no longer support flight, but they probably were not as well adapted to swimming as they are now, but they were still adapted enough to swim that they could function as swimmers. We could also see plenty of mammals doing this, like seals and walruses that have these funky little flipper things, right? That they're really, they look really silly walking around on land, but they get into water. They don't work as well as, like a whale or something, but like they do a pretty good job. And polar bears are another good example of this, because these big giant paws that are, they're walking and they're weight bearing. They're also really good at swimming with those things, right? So I don't know what this like fin leg thing is going for.
We're talking about two different things, right? So I'm talking about a one body part turning into a completely different thing.
Yeah.
You're talking about a wing going to flying to swimming. I'm talking about a completely different function. And there's a transition stage between swimming and walking.
Mm-hmm.
And there's going to be a middle part there where the leg doesn't work because it doesn't have the ligaments, the tendons, it can't support the weight, but yet it's too skinny to be used as a fin in the water. That's not the polar bear.
Are
with
you
a,
familiar with where are lungfish and mud skippers in this continuum? Are they on the good enough to swim but not walk, or good enough to walk but not swim side of the line?
they still have fins.
Right, but they can hop around on them on land and locomote
Exactly,
on
exactly.
land. Right,
That's my point exactly.
right.
That first fish would have looked like that and evolved a leg. That fish we see today
Mhm.
would have outcompeted that fish with that partial leg, partial fin. And that partial leg, partial fin would have died off because they couldn't swim and they couldn't walk.
So why are they still alive today? Why are there still mud skippers today?
Because they didn't evolve a leg.
Why haven't they been outcompeted? Why haven't they been outcompeted by the good swimmers and the good walkers?
I'm saying the mud skippers are the good swimmers and walkers. I'm saying the evolution part where
They're
a
the
mud
good ones.
skipper
Okay.
is turning into a leg, that transition stage where that leg is not able to support weight,
Are you
not able to
how
swim.
would you describe the bone structure of the lobe-finned fish? Let's look at them. How would you describe the bone structure of their pectoral fins?
I know it's shoulder-like, right?
They have a humerus, an ulna, a radius, and carpals.
Right, but that doesn't.
Right?
But you can't just.
They have all the bones that you need. They don't have digits. They don't have phalanges, but they have the rest of them. So you don't need all this new musculature. You don't need all this new skeletal morphology. You have bones that are already there. They're already connected to each other. All you're doing, if you look at like a coelacanth forelimb compared to a mud skipper forelimb, compared to a frog, a cat, a giraffe, a human, a bat, a whale, all you're doing is changing the proportions of the bones, right? Some are long and skinny, some are short and fat. You're changing the proportions of the bones within the limb. You're changing the number of bones at the distal end of the limb. That's what's different between these different structures, but it's not a whole bunch of new stuff that needs to be made. And the signals that allow you to have these different structures, it's similar to the folding in signal. There's only a very few developmental kind of signals that we use to generate all these structures. There's fold in, there's extend outwards from the midline. And the cool thing about the extending outwards from the midline, I want to shout out Mike from Answers in Atheism for teaching me about this stuff, is that it's when the extend signal, when it goes for a sufficiently long time, the tissues at the very end, I guess it has to do with the strength of the signal, the concentration of the chemical signal, they'll actually branch in almost a fractal pattern. So you get humerus, you get ulna and radius. Sorry, you can't even see my arm here. You get humerus, you get ulna and radius, you get carpals, metacarpals, phalanges in kind of a branching pattern. And we can demonstrate this very easily with just a very few chemical signals. It's not a big difference to get from that original lobe-finned morphology to the various terrestrial morphologies we see. You don't need all this stuff. We get like...
But you still have the lobe-finned fish with a fin.
Yeah, it swims.
Now you've got to get it to a foot, right?
Right,
That means
right.
you've got to lose the fin part.
No, you don't lose anything.
It
changes.
It's going to shrink down to a foot.
No.
Well, it has to. Look at a lizard foot, a leg, right? So what I'm saying is that you've got that radial foot, which also can paddle water.
Right.
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 Now think about losing. Now you've got this partial fin because you're narrowing down to a leg and a foot. It doesn't happen overnight, right? It's going to gradually give it less ability to swim and support its weight. That's what I'm saying. Even though it's got a shoulder, it's got all that, it's still going to lose function in that transition. And that transition is going to take hundreds of thousands of years, maybe more.
Yeah,
right?
Yeah. Oh, these transitions undoubtedly take a long time. Yeah. But
don't you think that fish with the 5 radial fin with the support and the ability to swim is going to outcompete the one who's now shrinking down to 2?
Why are we assuming it would shrink down to 2? I don't think there's evidence to support that position.
To form the shape of a leg, right? You've got to lose the radial fin. If you're going to put a foot, claw, and toes on a reptile, as what they say is the fish developed a leg from that fin.
Are you familiar with are you familiar with the limb morphology for things like a coelacanth and like a mud skipper and Tiktaalik? Are you familiar with the limb morphology for these? And there's a couple other ones in there too. The Acanthostega is another one. Are you familiar with like the bone structure for these things? Because it's not what you're describing.
Let's get down beyond the femur and the left. Let's get to the very bottom where the
the digits, right?
This is all we're talking about, right? Having that radial fin wide allows that thing to swim and support its weight, right?
Like a mud skipper, yeah.
Exactly.
So now, why can't you? So what I'm saying is, why can't you go from a swimming fin to a mud skipper fin without first losing, without having an intermediate step where you're missing, right? It's a structure develops in one morphology, and then you change the developmental regulation of that structure changes, and it has a different shape, but you're not losing any. I'm sorry, I'm not understanding it. You're not losing anything here.
No, we're still talking about two different things. Now imagine the leg of a reptile from hip to toe, right? You've got a foot, claws, toe, skinny leg all the way down, right?
Not really.
Compared to a radial fin.
Well, okay, so when you say radial fin, do you mean like a ray fin? Like a goldfish fin?
The rays at the bottom that touch the ground.
Okay. Yes. Okay, but like
what can use like flippers in the water, right?
Okay, but I need to be clear though, because if you're talking about like a ray finned fish where it has like like a fan, right, with the
at the bottom.
That what we're talking about?
Yeah, mud skippers have those at the bottom.
Okay, but but that's different from like the ray fins that like goldfish have. Those are separate types of fins here. The ones that mud skippers have, you've got these rays, but they're attached to a humerus, an ulna, and a radius. Exactly. Right? So like
we're on the same page now.
Good.
Now we have to turn the rays at the bottom fin into a foot to be a reptile.
Okay. They're already a foot in a mud skipper.
I know, but does it look like a lizard foot?
If you get rid of the webbing, it kind of does, yeah.
Exactly. That's okay. Now we're on the same page.
Right.
Now we're talking about evolution. Get rid of the webbing.
Right.
Now no longer can swim
because it spends most of its time on land.
It loses ability to swim
because it
doesn't have the ability to swim in the water because it doesn't have the webbing.
Well, it has the ability. It's just not as good. It's not an on-off switch.
Exactly. So
what's it better at?
Those can do better are going to outcompete it. So
but what's it better at?
Comes in.
What's that same organism better at though?
Hunting, swimming. It's going to have more support on the land because it still has both.
Exactly.
But the one that's evolving into a foot is losing. It's losing the webbing.
So what?
And it's losing these. It's going down to one. So we're going to be at a transition stage where there's no webbing. There's a small.
Why does it have to lose? Okay, so first of all, why does it have to lose the digits?
The rays that touch the ground.
Yeah, it has, but it has like other bones in there. You're assuming like it's not that it's just going to walk on little stubs.
The rays are what holds the webbing, right?
But the other bones elongate.
But
it doesn't just stay short and stubby.
They have to get down to one like a shin with a foot and an ankle.
Because that's never going to be. No, it's not. First of all, that's always two, right? The shin, there's two.
What I'm getting
and
at
then.
What I'm saying is that you look at a lizard foot and a lizard leg. There's nothing about it that's similar to a fish.
Who are they? Okay, so once they're on land,
they're not.
I think I see
for hundreds of thousands of years with a partially built leg and the webbing gone. They can't.
Who are they competing with?
They're competing with the other fish in the water that have.
But they're now adapted to land. Who are they competing with on land?
So you have to assume under your scenario that every single fish is evolving at the same time, the exact same way.
No, I don't.
Okay.
No, I don't.
Otherwise, the ones with the better fin that can swim better and still walk on land, now they're going to outcompete them for food.
I don't have to assume that, and that's exactly what happened. And that forced the ones. No, listen. Okay, look. That's it. What you're describing is a thing called niche partitioning, where you have a population where some members are better at one subset of the niche, and other members are better at exploiting a different part of that ecological niche, and they end up diverging into two separate species. So you have species that are coastal, where some of the members are slightly better adapted for dealing in water. Some of the members of that population are slightly better adapted for being on land. Competition is a lose-lose thing. Even if you win the competition, you have to spend energy to win the competition. So what you see in situations like that, it's called the competition dispersal trade-off, where there's a cost to exploiting resources that you're not as well adapted for. In this example, getting your food from on land. But there's a bigger cost in many cases to competing. And the question is, where is the larger cost? If there's a larger cost to competing, especially if you're losing, then you're going to disperse. And what you're describing is, so let's say it loses its limb, like its limb becomes this terrible, stupid thing where it can't swim and it's not good at competing on land yet. But that doesn't matter, because there's nothing on land to compete with if this is the first thing that crawled up onto land. So it can be not very good at competing on land, and it's going to be fine because it's the only game in town on land. If it tries to go back in the ocean, it's going to get its butt kicked. Who cares? I got all these resources over here on land that nobody else can use. And now you're saying, well, they all had to evolve at the same time. No, absolutely not. This is all happening a lot, and they're competing with each other on land. And only the ones that are best adapted on land are ultimately going to win. This is like a well-understood ecological phenomenon.
So in your scenario, here's the thing. You've got two fish, both have the ability to get on land equally, as far as one. They don't have one, whatever. So both are going up on land and back. So now you have a partial leg fin that's weak, that doesn't swim and is not able to support weight as well. So now you've got something that's not as good at land or water. So
but there's
neither the one that can still go up on land with the fin can still do it better than the one with the leg that doesn't work.
Okay, so but that so what are they competing with on land? Right? They're competing with each other, right? If multiple different groups or multiple different individuals are doing this simultaneously and they're interacting, they're competing with each other. So who's going to be the best at being on land? The one that develops the best limb and the best lungs fastest, right? They're going to be the best. That genetic profile that leads to that developmental pattern that gets you the best combination of limbs and lungs, they're the ones that are going to be best adapted for land. And are they going to care what's going on in the ocean? No, they're not going to go back to the ocean because they're the best ones on land. Like, you're describing exactly how this would take place and then reacting with incredulity of like, how could that even happen? But like, you're describing the process of how it would happen.
No, what I'm saying is, let me put it in an analogy. Okay, got two people. Okay, one, and they both go in the water and land. One of them has a small fin that they use in the pool, right? So they can swim. The other person has a cast on their leg and can't hardly walk because they broke their ankles. Now you tell me if the guy with the fin that can swim in the water and uses fin on land is not better at both. That's the scenario I'm saying happens when you have a non-functional stage where neither one works.
So two things. You are assuming a non-functional stage that is contradicted by one, organisms that exist today, and two, the fossil record. So I just want to make that perfectly clear that you're building into your argument something for which we have no evidence. But the second thing, and the more important thing, is the underlying evolutionary processes of this process. It, like in your hypothetical scenario where you have something that can barely walk and barely swim competing with something that can both walk and swim, the thing that can do both things well is going to win that competition. What I think you're missing is that that thing is going to have a lot of competition in the ocean, competing with lots of things that can swim very well, but it's going to have no competition on land from other things that can walk very well. So it's going to end up getting its resources on land, and that is going to impose a selective pressure to get better and better at being on land. Within that land capable, I'll say, that land capable population, what you're doing is imposing selection to emphasize those land adapted traits. And like, this is very straightforward in terms of tetrapod evolution. We have a really robust fossil record of this stuff because it happened in coastal regions where fossils form really easily. Like, this is, again, this goes back to what I said earlier of like the incredulity side of it. You don't have to believe this is what happened, but like, we have a lot of evidence that what you're describing is what happened. And now with genomics in the field of Evo Devo, we can explain the genetics of it. And just like we don't need new bones and muscles to get these new limbs, it turns out we also don't need new genes to get these new limbs. The genes that govern tetrapod limb development, it's the same regulatory network that governs limb development in arthropods, in insects. You can look, it's like a one-to-one match with these regulatory networks. You don't need a whole bunch of new stuff. It's just about the development pattern and gene expression patterns and regulation during development. Couple that with selective pressures that push you in one direction or another, and there's your adaptation. There's your new limb.
I'm gonna give you the reason this came up to me is I'm gonna give you a paper. I don't have the name of the paper, but I read it, and it was these scientists. They found this archaeological record. They found all these fossils, and they said this is how a lizard jaw formed into a mammal jaw, right?
Oh yeah, the jaw progression, yeah.
Right. And so I read through it. And so it was like this led to this, led to this, this. And in their own paper, they said, we get to this stage here, and the jaw is non-functional. Now, how we get around that, we have no idea, because it's non-functional. And then they just skipped on to then it happened to this and this and this. So they in that paper said exactly what I'm saying right now. There's always going to be a part where it's non-functional, and it's in that paper. But yet the evolutionists saying, hey, this is how the jaw formed from this and this and this, and this is how it happened. Look at the fossil record. But in their own paper, they admit they have no clue. So
I am not an expert on the reptile mammal transition, but I happen to know somebody who is. So what I want you to do is look up the book called Evolution Slam Dunk by R.J. Downard. I guarantee you the answer to whatever that question is. I guarantee you it is in that book. That is that's the source for like.
This came
look it up.
This came from the guys that actually drew out every single fossil and showed the exact, this is what happened, this is what changed here, the inner ear moved here, and then we get to this part of the jaw where it's non-functional. And they said that right in their paper.
So you'll have to forgive me for not exactly taking your presentation of this paper at face value without the direct citation of the paper, given the I'll say the well-documented history of misusing papers in anti-evolution contexts. I would love to see that paper if you could email it to me once we're done at some point. That would be great.
Yeah, that's what really struck me, as there's always a stage where something doesn't work when you're developing a new body part from an old one.
I think you're getting way out over your skis by saying always in that sentence. There is going to be a stage necessarily where you're developing a new feature. If you're accepting an old feature to do that thing, in most cases, you're going to lose the old function at some point. In some cases, you don't have to, but in many cases, you will. That doesn't create a barrier to evolution of that.
The barrier that it creates is that it stops the reproductive advantage because it limits mobility, or whatever the trait was it had before isn't developed into the new one. It limits, and so it doesn't increase reproductive, it decreases. So it's not going to generate that into the population. Now, I don't know how, like with the fish leg and all that, I understand all the things you're saying, but I've not heard an explanation of how, if the fish that can go inland and water can outcompete the one that can't do both very well, but still can outcompete it in both land and water, how does the one that doesn't do it well increase its genetic throughout the whole population when it can't?
That's the answer. It doesn't. The one that does do land well wins that competition.
What I'm saying is
that's the point.
You've got a fish that does not do land well yet because the leg doesn't work, and doesn't do water well at all. Doesn't do well, doesn't do either well.
Yeah, that one's going to lose if it has to compete against something that can do land well. That one's going to lose.
Exactly. That's what I'm saying. The one with the fin never changed can do land better, and it can do water better, because the other thing can't swim well and it can't walk well. Because it's,
I think you're. So I don't think you're correctly representing the fossil record on this question. Going back to the limb morphology rather than the jaws. But again, I want to emphasize for everybody that, again, we got an example of I don't see how it could work, which, like, fine. But like the ecology of that process I described earlier with like niche partitioning and the competition dispersal trade-off, like these are really well documented things that we can like quantify. Like we can actually like show how this works and how using different parts of the ecosystem are going to lead to different outcomes, better or worse, depending on how you're competing. Like we're not just like sitting around like, oh, I bet this is how it happened. Like do you understand that this is like a robust, quantitative, well-developed field where you may not like be on board with these explanations, but like, we're not just like sitting here in our dorm room BSing. Like we do actually have answers to these questions that I'm trying to convey here.
Well, so far, I haven't got an answer.
Okay.
You've gotten, we know how to do it. We know how to document it, but I haven't gotten an answer. Like I just asked you the question, if you're going to use evolution, you have to use the laws of evolution, right? And
what are the laws of evolution?
That means reproductive advantage.
Is that the only process of evolution?
That's what else is there?
Genetic drift, horizontal gene transfer, non-random mating, gene flow, mutation. There's a lot of mechanisms of evolution.
In order for
genetics, niche construction.
Right. In order for a set of genes to become widely into the population, it has to be reproductive advantage. It has to outcompete.
That's not true. That's not true. That is 100% not true. Because no, that's not true. Nope, that's not true. It's not true.
Then how's it
you can have things reach fixation by genetic drift. You don't need to have a selective advantage to reach fixation. That's especially common when you have a hitchhiking effect, where a neutral allele is linked on a chromosome to a highly beneficial allele. So you don't need to have a selective advantage for a trait to reach fixation within a population. And no, Glenn, this is where I have to say what has been kind of gnawing at me for the last half hour, which is this. You come in here, you like, for a long time, I've been very patient with you commenting on my channel, being very rude to me and other people. And I've given you more leeway than I've given other people. Very polite via email to set up this conversation. We've been chatting for well over an hour now, and I'm having a grand old time. But you come in here and tell me that, like, well, what other mechanisms are there? The natural selection. I like do better than that. Like, seriously, do better than that. Like, you're going to come in here and say that I don't understand how this can work. There's no explanation for this. And you come in here and you couldn't pass a midterm in an intro evolution class. Like, what are we doing here, frankly?
That's what I'm wondering. I had these questions, right? And so far, I haven't had answers. I've heard a lot of theories and hypotheses, and this is how we do this and how we do that. But even the fish leg thing, you can't explain to me how one thing would outcompete the other and take over the other one.
I provided you the mechanisms for it, and I encourage you to look up. There's a lot of really good resources out there. I encourage you to look up processes like niche partitioning and the competition dispersal trade-off, which really does explain how a lot of these things work. And you combine it with the evolutionary genetics and the developmental genetics of limb development, which again are consistent not just in tetrapods, but across animals, right? To the point where the regulatory networks are the same between like mammals and fruit flies, right? We have an understanding of the genetics. We have an understanding of the ecology. Like, I don't know what more you want for that. You picked a question where we have an extremely robust understanding of exactly how that works.
How about, since we're talking about evolution from a common ancestor, we have to add DNA, right?
Yeah.
How about an example of that? A new gene.
How many do you want? We've got fish that have de novo antifreeze proteins that don't exist in anything else. We've got bacteria with nylon digesting enzymes that don't exist in anything else. We have a bunch of genes that chimps don't have. Like,
so the nylon, right?
Mm-hmm.
That the same people that did that research went back and they found out it was not a new gene. That's been published.
It wasn't a de novo gene from a frameshift mutation that conferred a start codon was the initial hypothesis. That's not what it was, but it is a new enzyme. It's within a family of enzymes that digest nylon-like molecules, but there's no other enzymes outside of those specific things called nylonases, right? Those things are part of a larger group of enzymes. They are new. They did not exist until the 20th century, right? That's a new gene.
So this is where, I hear a lot of these things about this, okay? So let me just read you their findings, right? Real quick. Where did the nylon eating ability come from?
Who are you? First of all, who are we citing here?
This is from the two scientists that initially did the research.
Do you have their names?
The Japanese guys.
Yeah. Okay. So the original hypothesis was that it was a frame shift that conferred a start codon. That turned out to be wrong.
This is
right.
Before they just assumed that it was from that.
What year was that?
They found out.
What year was the original, please?
I don't have the paper.
It was in the 70s.
Yeah.
It was in the 70s, and they corrected the hypothesis, and we now know the correct answer to that one.
Their binding pocket is large, and it can accommodate a lot of different substrates. They are promiscuous enzymes. Yep. In other words, the carboxylyesterases reaction hydrolyzes a chemical bond similar to the one hydrolyzed by nylonase. It was just generic.
So there was an ancestral enzyme that could catalyze a reaction for similar molecules, and then in some types of bacteria, you ended up with a new gene that could catalyze nylon. Yes, exactly. That's how we get new stuff.
There was no new gene.
It's a gene duplication, and then one of them acquires a new function.
L and L did not have a frame shift new folds. They had a pre-existing folds with activity characteristics of their old fold. There was no brand new protein, no novel protein fold that emerged, and no frame shift mutation was required to the nylonase.
All of what you just said is correct. That doesn't change the fact that it is a new protein, nylonase, that did not previously exist.
But it said it could. It was an easy thing for it to do.
Yes, it's not hard. It's surprisingly easy to evolve new proteins. I don't know why.
It didn't create a new gene to do it. That's my point. It's the same with citrate.
You mean in the Lenski thing, the citrate? Right. It copied the module, so it had one copy that was anaerobic and a new copy that was aerobic. Yeah, but okay, you want more? Okay.
No, okay,
Well,
exactly.
how many?
I'm just saying these are the ones I hear all the time about a new thing, a new gene being created. The Zachary Blunt did his PhD research on the citrate,
That's a gene duplication.
and he
That's new.
said
That's
what he
new
found.
information.
There was a step that involved at least 2 mutations. He identified one likely mutation, a single nucleotide change that damaged a gene, which resulted in an upregulation, so gene degradation.
So that's referring to the reimportation.
A duplication of that gene
Different
that
gene.
produced the transporter protein.
Right.
And that was just moved. A promoter was moved.
No, the promoter, that's wrong. The promoter was not moved.
This is what he got his PhD on.
The promoter, no, you're reading outdated. No, Glenn, you're reading outdated research, Glenn. The promoter was not moved. The gene was moved. It was a copy. A gene was duplicated into a region controlled by a new promoter. Again, you come in here, you come in my comments with rude comments. You come on here with outdated research. What are we doing here?
This is March of 2016.
Still outdated. It's not. That's what you just said was wrong. It was not a promoter duplication. It was an entire gene module was duplicated into a region controlled by a different promoter.
But it started with a gene degradation.
No, it didn't. You're calling a mutation a degradation for no reason. Yes, it mutated. There's actually multiple mutations in that system. It changed a gene, didn't damage it.
It resulted in an upregulation. Right?
Gain of function, yeah.
Which enabled a rapid metabolism of citrate.
And then yes, and that's a new trait, yes.
And that's not a new gene, then.
It's a gene duplication. It's literally something that wasn't there before.
Duplication is just a copy.
Right.
Right?
Yeah.
I have 2 keys to my house. They do the same thing. Is that a new function?
If you go to the hardware store and make a copy of it, then yes, it is. Especially when one of them is used on your front door and the other one's used on your back door.
Then that's not a copy.
Well, these two genes that were copied, they're expressed under different conditions. They're not doing the same thing. It's not just doubling the activity. It's 2 different sets of things.
Then it's not a copy.
Okay, then it's not a copy. It's a new gene. Like, pick one. It's one or the other, right? Okay, look. Do you know that there are dozens of de novo genes just in humans compared to our common ancestor with chimps, and we know where they came from and how they got that way?
See, that here's the thing is all.
Well, please just answer that question though. Are you aware of that fact?
I am aware of that. Yes.
Okay, good.
But that still didn't explain how all of the new DNA built us.
We do know how those genes got there. You have non-coding transcribed regions that acquire a start codon. So you get what are, in some primates, they're long non-coding RNAs. They're transcribed, but they're not translated. And in humans, they've acquired translational ability. So we get a whole bunch of de novo protein encoding genes through simple point mutations where we acquire start codons, and those previously non-coding RNAs become protein coding. And then those proteins will have activity, which can then be refined through natural selection. I do just want a programming note for everybody. We've been going for 1 hour and 23 minutes. 90 minutes is about the limit for these things.
Okay.
So that's what we're gonna, so let's bring this home. We've been working on, so a lot of this has been irreducible complexity. So Glenn, take it away.
I didn't want to come across as angry or whatever. I appreciate your time.
No, you're good. You're not coming across as angry at all.
What I get frustrated with is that you read something from one side that makes a claim, you read something from another side that says, no, this is not how it happened. And there's not an actual set that an agreed upon for anything. And when you can't get to the point where you can't figure out how a cell even started, or how did mitosis evolve into meiosis, the actual process of that, that jump, things like that.
If we had another hour, Glenn. I know that you're going to talk about the algae and all that, but that's not mitosis and meiosis. Right? No, I wasn't going to.
From single cell to a sexual reproduction.
No, I wasn't going to talk about that. They already had meiosis at that point. The unicellular Chlamydomonas has meiosis. That's earlier. You got to figure that out earlier.
But it's still a whole different process.
Yeah, it is. It's really cool how it evolved.
Yeah, but I'm just, it's all a lot of theories and guesses, and we see a virus that did this, so we can extrapolate that that's how the human mind was built.
Nope.
That's the way it's read in the papers.
That's not how it works. No.
Papers, a lot of the papers you read, there's always we think, we assume, we this or that, but there's never, it's proven this is how it happened, or now we know.
That's actually a really great point, because what you just hit on is the difference between science communication and regular conversational communication. So in a science paper, when you say, we assumed X, Y, and Z, what they're describing are the, other people have done this, right? So what they'll say is, well, we assume the conditions were X, Y, and Z, and then cite somebody that found those conditions, or, whatever, just to use an example. When they say, we propose or we think, what they're describing is their findings in tentative language, because that's how science writing works. That's how you state your conclusions when you are proposing them in scientific writing. You say, we observed X, Y, and Z. This is an unanswered question. We did blah, 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 blah. And we propose as a solution, bing, 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 here's why, right? And the language is necessarily tentative. That's how science writing is. That doesn't mean we're just guessing. It means here's what we're putting forth along with our evidence that supports that idea.
Right. I'm not saying it's guessing, but I'm saying there's always a caveat of.
There are lots of caveats, always. Yes.
We're not sure, right? We're going to make an assumption because we're not sure, and then we're going to make a conclusion. There's a lot of that.
So that's where, so it's not, we're going to make an assumption, we're not sure. We're going to, based on what we know, for example, abiogenesis research or something, based on what we know, here are the conditions, right? We're going to test these conditions, those conditions, whatever the case is, right? We're going to assume the conditions were X, Y, and Z and see if this process could happen. That's not saying we're just going to assume in a metaphysical sense, right? It's for the purposes of this experiment, right? There's always caveats. There's always specific conditions that we're operating under. That's how experimentation works. That's a feature, not a bug.
Yeah, specifically, when they're talking about gene duplication, right? They describe the process, which is known. They can figure it. They can see it. They can do it in the lab, and then they say, "This is how we propose that genes were added to this, this, and this," but they don't know. They don't know for sure. They just say this is the process, and then we assume this is how it happens.
But we can say, look, when a gene duplicates, we see these things happening. Oh, look, here's a gene family that has all these signs of gene duplications, right? We can make predictions about, we know what gene duplications look like. Yeah. We know what that looks like. We can do that in bacteria and virus. We know what that looks like, right? So now we can say, okay, or even in eukaryotes, we can do it in the lab and see it happening. So we can look at that experimentally and say, okay, we know what the results of this process look like. Now let's look at the mammalian genome and see if we can find signs of gene duplication. Or let's look in the chordate genome and see if we can find signs of whole genome duplication, because that's another thing that happens. And we can find 2 instances of whole genome duplication within chordates. One from the invertebrate to vertebrate transition and one from the jawless to gnathostome transition. Right. Full genome duplications, right? We're not just saying oh, this might have explained it. We know exactly how these processes work, what they look like, and then we can make affirmative predictions about what we should see in our genomes if these processes actually happened in specific cases.
So I didn't finish the part about, they say a replication, and then that new gene magically mutated into this new thing.
Nobody says that.
Yes, they don't say magically, but they say this is how it happened. There's a gene duplication, And then that new gene mutates into something new and creates a new protein.
Yeah.
And that's how we assume it happens.
But we don't assume. We can document that process.
I've never seen it documented. That's what I'm saying.
I can give you some references.
I would appreciate it. Like I said,
So it is
I open mind. I'm just saying. I hear a lot of times that it's a done deal. It's proven and all that, but I've never actually seen like the things I'm asking about. Like I've never seen a gene duplication and a new gene and a new function. If you can send that to me, I would love to see it. I mean that in true, honest.
Okay. Well, you'll have some emails coming your way then, because I got you covered.
Thanks.
So we're at 90 minutes. So let's take a couple minutes and wrap up your where you're at, Glenn, and then we'll bring this home.
Okay. Well, I just, obviously you're way more educated than me, but there's some basic big things like the formation of the cell, the DNA, the structure of that. And I still don't think it's as easy as you say for, new proteins to be made and directed by the developmental proteins in coordination blindly. I just don't think that's as easy as you make it sound. But that's my takeaway or whatever from this. But I really appreciate your time.
Okay.
Thanks so much.
So for my money, I think a lot of this stuff has been really well explained. And I want to suggest everybody, if you're looking for a good intro level evolution book, Zimmer and Emlyn, Evolution, Making Sense of Life. Good book. Covers a lot of this stuff. I like that one. I always recommend that one to my students. That's a good one. I think for everybody watching, I think this has been a really good example of what we often encounter in these conversations, which is the kind of the argument from incredulity kind of side of things, where I'm skeptical that there will be sufficient evidence that I can provide you that will convince you of any of this stuff, but I'm going to try anyway. So we'll keep chatting, I'm sure.
Yeah.
And I do want to thank you, Glenn, for coming on. I really appreciate it. I love having these conversations. And I also want to just so everyone, if you didn't hear me say this at the top, Glenn is a common commenter on the channel. And what I always do is say, like, if you got a problem, like, email me and we can talk about it. And that's when most commenters just stop because nobody's really willing to talk about it. But Glenn, to his credit, was like, yeah, okay, let's do that. Let's set it up. So here we are. So I really appreciate your willingness to do that. There are not many people that are going to do that. So thank you. I'll be emailing you. And if you could make sure you could send me the papers that you have as well, I would really appreciate that. And that is where we're going to leave it, everybody. So thank you all for watching. If you haven't already hit that like button, go ahead and do that for me. I would appreciate it. And we'll have more stuff coming out on the channel next week. We'll have some stuff. I'm reading a book coming up. I just did a book review, but I got another one. I got Glass House by Ken Ham and Bodhi Hodge and a bunch of other people. So that'll be at some point over the next few weeks. And that's where we're at. So we're going to call it there, everybody. This was a little longer than the usual conversation, but I think it was fun. I hope you all enjoyed. Have a great night, everybody.
Thanks, Dan.